Hello, welcome. This is Mr. Watkins. This is another video lecture. This one will be on DNA replication. You should have already watched the structure, or at least have the knowledge about what the structure of DNA is before you go through this video. So let's get started. Again, these materials can be downloaded off of MrWatkins.com. So here is our DNA strand. Remember, it is a double-stranded molecule. It's made of subunits called nucleotides. The nucleotide consists of three pieces, a phosphate group. In case of DNA, this sugar, this 5-carbon pentose sugar here, is going to be deoxyribose. And then it has 4 bases that it uses to form this sequence: thymine, cytosine, adenine, and guanine. So those are the 4 bases. The bases are divided up between whether they are double-ringed or they are single-ringed, and that there is a base pairing rule, a complement that each of these bases will bond with. Also remember from the last video, or from the video on nucleotide structure, on nucleic acid structure, the DNA runs in opposite directions. It is anti-parallel. So let's talk about the strand real quick as far as its direction. Remember that the carbons are labeled, and that this carbon here is C1, and this carbon on this end is C5, and then this one here is C3. And so when we look at this molecule, what we're seeing is the 5' end on this strand, because we have number 5 here. So this is the 5' end here. Now on this other strand, we have our 5' end up over on this side, but our 3' end, our carbon 3 end is open, so this is the 3' end. And as you take this molecule and you follow it down and follow it through all the way to the back end here, you're going to see that our C3 end is open, so this will be our 3' end. And then on this other strand, our 5 carbon is basically open, so this is going to be our 5' end. We're going to read this molecule in this direction, and we're going to read this strand of the molecule in this direction. Before we get started, let's go through and do our complements here real quick. So we have thymine. What always bonds with thymine is adenine, so there's our adenine. Cytosine is always to guanine. Adenine to thymine, and so on. So I'll get those cut and put in here, or excuse me, written in here real quick. And I'm just writing the letters into the spot on the actual base. And cytosine, guanine, thymine, and adenine. Okay, so double helix. This strand, it is normally wound up, curled up in a double helix, in a helical pattern. So the first thing, and again this is for pre-AP, the first thing that needs to happen is that the strand needs to unwind. And when you get to AP or IB, you'll talk about the name of that unwinding protein, the one that holds them down. And then the next thing is this bond here that forms between these bases needs to be broken so that we can get in and make a copy of that sequence. DNA replication is to make a copy. You get a cut, or you're growing, you need to make more cells. Each of those cells has to have a copy of their DNA, so that they so that they can make all the proteins that that cell's required to make, to be part of that tissue, or whatever, or even just that organism. So we're gonna say that we need to come in and break this bond. So for this model that we're using, we're going to use scissors to simulate that particular enzyme that comes in to break it. And that enzyme that you need to know, and we're going to keep an enzyme sheet here, is called Helicase. So let me flip over to a blank piece of paper so that you can we can keep track of the enzyme. So this is going to be DNA replication enzymes. And our first one is Helicase, that you have to know of. And what does it do? It breaks the hydrogen bonds between the bases. Sometimes we say the DNA has to be unzipped, just like you would unzip the zipper on your jacket or a coat. it has to be unzipped. So Helicase will be our first one that we are going to simulate and we're going to look at here. So let's go ahead and bring our molecule back in and let's do that simulation. And here's my Helicase, and I'm going to come in and break this bond and just going to cut right through it, right through the bond. Helicase splits this open so that the other enzymes can come in, and I'm just going to cut up through that first page so the enzymes can come in and make a copy of that particular molecule. So our helicases come in, it's separated out, and then the next step is, or it's separated out, so let's go ahead and separate it out, and then we're going to come in and make a copy of that DNA. So I'm going to take a little bit of tape here and going to tape down one side of the molecule here, and then I'm going to move the other side over so that you can get an idea of what's going on here. So we have this separation here. Now the next thing that's going to come in, okay, so we've had it split open, and one little thing I forgot to mention here, this part where it's split right here, okay, that is, this whole thing is called a replication fork, and that part where it starts is called the origins of replication. And so right now we're going to go ahead and add in our nucleotides. We know that thymine, its complement, it always bonds, its base pair is always going to be adenine. Now I've pre-printed and cut off the bases. I did it on a different color so you could see them being added in. And it's going to, again, add in, and it's going to add in basically upside down. Now we have our 3' end here, but that means on this molecule here we've got our 5' end. And we've got our 3' end showing. Now in AP and IB there's going to be some other molecules that have to occur before this process actually has happened, but in pre-AP, you know, there is a primer that's put down that gets it set up so that our next enzyme can come in and do its job. Our next enzyme that comes in and adds in these nucleotides, this next enzyme is called DNA polymerase. Now DNA polymerase can add to a primer, so we're going to assume that it's already ready to go, and the DNA polymerase will come in and it will add a nucleotide. So we'll create that hydrogen bond to the complement, or it'll add the complement to the strand, and it'll leave this 3' end open. DNA polymerase can only add to its complement as long as there's a 3' end available on the new strand that it's making. And in this case, we're going to say the primer was here and had a 3' end. We do have a 3' end here, and this is the only way it can add. So let's go ahead and add that adenine on. So there we go, I've added it in. Again, this is my 5' end, my 3' end. The DNA polymerase is going to shift down. Adenine's complement, its base pair, is going to be thymine, so this thymine will come right on in and DNA polymerase will add right to that thymine. And it can add to that thymine as long as there's a 3' end available on this end, and there is. And what will happen is a bond will form. I'm just going to put a little piece of tape here so I don't write on the paper underneath. The bond is going to form from, again, remember, we've got our, between our pieces of nucleotide, that bond is going to form with that number 3 carbon of that sugar. We still have that 3' end open, and again, DNA polymerase can only add to that 3' end. We're going to have to form this bond. So here's our adenine, our complement is thymine, so we're going to add our thymine in, and DNA polymerase will get that thymine added right on in, and we'll also have to form that bond to join this growing nucleotide sequence together. So I'm just going to put a little piece of tape here so that I can join it up. So here we go with our phosphate over to the 3' end. So we're building that bond. We're building up this sequence. There's cytosine. Its complement is, it is guanine. So we're going to add our guanine nucleotide. If I can pick one up here. Got our add our guanine. Again, we still have a 3' end available, so we're going to be able to continue to do this and to continue to add until we get to the origins of replication. We get to where that spot on that replication fork where Helicase will have to come back in and open it up some more. Again, always remember that DNA polymerase can only add as long as there is a 3' end to add to, and it's got a complement on the other side where it can add. I'm going to move this down just a bit so that we can continue to add here all the way up. So I'm going to put this down so we can continue to add. And there's a cytosine that's going to be added in here. So we're going to go ahead and put the cytosine in. And cytosine, we also have to make our bond between the nucleotides, between the phosphate and that C3 carbon on that sugar. And we need another cytosine here. And I think you're getting the idea of what's going to go on here. And this side, we're going to have this continue to add until it gets to a point where it just can't get in there and add anymore.